Greetings, students, and welcome back to the last part of our notes for our current unit, Chapter 8. Today I want to first finish off what we started talking about the other day, which was meiosis. I want to talk a little bit about how meiosis assures genetic variety and a couple of different processes that take place during that process. First thing I want to talk about is something called independent orientation, or sometimes they refer to it as independent assortment. And this is a process that occurs in meiosis that really leads to potential for a variety of varied offspring. Because if you remember from what we talked about in the previous lesson, the chromosomes will line up in homologous pairs. And the random arrangement of the chromosomes pairs during metaphase 1 could lead to really many different possible combinations of chromosomes when you think about it. Depending on how, if they line up left to right or right to left, will determine ultimately later what daughter cells that they end up in. So that's one way variety is built in as a mechanism of meiosis. Another way is this idea of random fertilization. Let's say, for example, with the sperm, if you have 4 daughter cells, 4 sperm cells, whichever one is the one that actually fertilized the egg is going to be the one that passes on the genetic information. So 3 are not. So random fertilization will also increase this ability to pass on offspring that are varied or very different from the parent. So this is what independent assortment looks like here. You can see in possibility A how the chromosomes are lining up, blues on the left, reds on the right. But look what happens if this second pair here actually lines up red to blue instead of blue to red. Look at how it changes the daughter cells in comparison, and here and here. And then look at the daughter cells after it's all said and done. Instead of a blue homologous pair, red homologous pair, now I have blue and a red, blue and a red. So I have different combinations just by the way they lined up. The genetic information is the same in each one of these chromosomes, but how they line up will affect where they end up in their daughter cells. So even though the information might be the same in possibility A and possibility B, we end up with completely different outcomes just by how they lined up in homologous pairs. Now, the difference between homologous pairs comes from the fact that they have different versions of genes at corresponding loci, which means like you may have a set of genes coding for a particular trait, and your paternal genes have one set of code, your maternal genes have another set. So each parent is providing slightly different information for that particular trait. And in the process of crossing over, that's an exchange of corresponding segments between non-sister chromatids of homologous chromosomes. And so what does that mean? So it means basically during the early parts of meiosis 1, your homologous pairs will actually swap genetic information. So like this piece of chromosome will actually change with this piece of chromosome. So now each chromosome is actually definitely different than it was before because a whole section has now been replaced. So what was on the maternal side will now be on the paternal side and vice versa in that piece. So that's definitely going to assure some genetic variation. Okay. And this is an image of how they attach together. When they form the tetrad, you can see the sister chromatids, and you can see the tip of this one now has been changed. That maternal piece has picked up a paternal piece. Okay. And so all these things together increase genetic variability just astronomically. So we have, again, crossing over, which occurs during prophase 1, and independent assortment, which occurs during metaphase 1. And then, of course, throw in the random fertilization as well, and all those things will increase variation. And so this slide kind of shows you a couple of those elements there. You can see the genetic information has been crossed over here. You look here at the results. Depending on how they line up, we'll determine what ends up in the daughter cells. So great opportunity for some genetic variety. Now, the last thing I want to talk about in the chapter is the alteration of chromosome number and structure. So everything we've been talking about so far is under the assumption that everything goes perfectly. The genetic information is copied perfectly, everything lines up perfectly in homologous pairs and so on and so forth. But sometimes that doesn't happen right. An abnormal chromosome count is the result of something called non-disjunction. And non-disjunction can occur a few different ways. First, it could be the failure of the homologous chromosomes to separate properly during meiosis 1. Or it could be the failure of the sister chromatids to properly separate during meiosis 2. And so let's see what that looks like. So here's meiosis 1. Notice the homologous chromosome pair is separating here, but it's not separating here. So now we're ending up on this side a lot of genetic information, but on this side not enough. So each one of these daughter cells has one extra chromosome. Each one of these daughter cells is missing a chromosome. And look what happens in meiosis 2. let's say, for example, we have a normal meiosis 1, the chromosomes separate properly, but now the chromatids don't separate properly. So now we end up with extra chromatids on this side, okay? And a piece missing on this side here. So when those daughter cells divide, now this piece has extra, this doesn't have enough, but ironically, look at this line here. Those are correct. And this is where that random fertilization part comes in. Let's say that there was a mistake like this. There's still a built-in mechanism that could provide an opportunity for the proper genetic information to be passed on if either one of these cells is able to fertilize the egg. Now, how do scientists study chromosomes and homologous pairs and things like that? They use a device called a karyotype, which is a photographic inventory of an individual's chromosomes. To prepare a karyotype, blood cells will be extracted from the patient. They'll be separated, stimulated to grow, and then during metaphase, they'll capture images of those chromosomes and observe them under a microscope. And what they do is they arrange them into ordered pairs so that chromosome abnormalities can be detected. This is what a karyotype looks like here. You can see this is the mess of chromosomes, not unlike what we'll be doing in our upcoming lab. And the chromosomes are put together in homologous pairs, which are chromosomes, remember, of the same size and kind of content of what they share. The paternal and the maternal chromosomes put together side by side. And what they'll do is after they assemble them all together, look for any issues, damage to the chromosomes, missing chromosomes, extra chromosomes, that kind of stuff. See, there's 22 pairs of chromosomes. The first 22 pairs are called autosomes. And then the 23rd pair is the sex chromosomes. And these are the ones that determine things like gender. For example, XY would be a male, XX would be a female. This is just a kind of a graphic that shows you how they go through the process of getting the information ready to observe it underneath the microscope like this lab technician is doing, and you can see the building karyotype here. Here's another example of a karyotype here, and you can see 23 pairs here. So this is, of course, a human karyotype, obviously, the 23rd pair XY. So this is a male. Now, sometimes you have issues where there might be too much genetic information or too little. An example is trisomy 21. This is an issue with the 21st chromosome pair, the homologous pair there. It's sometimes referred to as Down syndrome, which I'm sure most of you have heard of before. This is what it would look like under a karyotype. Look at the 21st pair here. It's called trisomy because there are 3 chromosomes there where there's supposed to be 2. And so this is part of the upcoming lab we'll be doing, where we'll be analyzing these karyotypes and looking to see if we can detect any types of genetic disorders. And something interesting here, you see this graphic here that shows the relationship between the age of a mother and the potential of having an infant with Down syndrome. You can see it goes up kind of dramatically once the mother passes 40. Interesting observation. Now, abnormal numbers of sex chromosomes don't always affect survival. I mean, sometimes there could be exhibited disorders. Sometimes they can be substantial. Sometimes they could be very inconsequential and hard to notice at all. Sometimes you don't notice them at any way, shape, or form. Okay, and sometimes non-disjunction of sex chromosomes can result in individuals with a missing X or Y. In some cases, you can end up with an extra, like here. We have XXY. In other cases, you can end up with 3 X's, XXX. Okay. And so these are some of the abnormalities that you might see in sex chromosomes in humans. This chart will be part of what we'll be doing in the upcoming lab, where we'll be trying to diagnose some of these types of syndromes based on the chromosome information that you've acquired from building your karyotypes. Now, non-disjunction can produce what are called polyploid organisms, which are organisms with extra sets of chromosomes. And surprisingly, sometimes these errors can actually be important in the evolution and development of new species. And you're reading, they talk about the gray frog as an example of one that's a result of that. So you can read a little bit more about that in your text. Now, some other types of chromosome alterations that you could witness are these here, which are called deletions, duplications, inversions, and translocations. These can produce some genetic orders, things like cancer and stuff like that. And this is what they look like here. Deletion is, of course, where a piece of genetic information has been deleted. So now it's missing. So whatever it's been coding for in that particular chromosome is now missing. Duplication is where a piece of genetic information has actually been copied twice. So now the chromosome actually has extra genetic information. In inversion, you can see the genetic information is actually kind of flipped around, so the ordering is kind of out of whack. And then reciprocal translocation is actually where you have genetic information from non-homologous chromosomes, different chromosomes, exchanging places. So now we have information that's been swapped on different chromosomes. Okay? And all these can, of course, result in different types of genetic abnormalities. Okay. And this is a little slide that talks about some of the different types of common disorders, some of the different types of examples of genetic disorders, and those kind of things. And so that'll conclude our chapter. And in the next chapter, we'll look at more into genetics as we start talking about Mendelian genetics.